Bismillahirrahmanirrahim. Assalamu alaikum. Welcome to my channel Biology Expert. Today we will learn about the process of mitosis. Mitosis is the combination of two words, mitos means thread, osis state. So mitosis is a division in which the threads form, and these threads are actually spindle fibers. Due to the formation of spindle fiber, this division is called mitosis. Sometimes cell divides by nuclear constriction. No spindle fiber is formed. This type of division is called amitosis. Because there is absence of spindle fibers during this division, no spindle fiber is formed. That's why this type of division is amitosis. And mitosis is a division in which the spindle fibers are formed. Mitosis is a process where a single cell divides into two identical daughter cells. The major purpose of mitosis is for growth and to replace the worn out cells. Mitosis is divided into 5 phases. Let us discuss these phases one by one. The first phase is interphase, and this phase is also known as growth phase. And during this phase, the DNA in the cell is copied in preparation for cell division. This results in two identical full sets of chromosomes. And during this phase, the centromeres, centrosomes are also divided. At the start of this phase, there is only a single centrosome was present, but at the end of this phase, centrosomes also double. Outside of the nucleus are the two centrosomes, each centrosome containing a pair of centrioles. These structures are critical for processing of the cell division. In centrosome, two centrioles are perpendicular to each other. During interphase, microtubules extend from the centrosome. As shown in the figure, that there is the formation of spindle fibers. Small spindle fibers are arising from the centromere. And you can also see in the diagram that the chromatin material are in decondensed state. Chromatin material are in open conformation. The next is the prophase. During the prophase, chromosome condensed into X-shaped structure that can be easily seen under a microscope. Each chromosome composed of the two sister chromatids containing the identical genetic information. Chromosomes pair up so that both copies of the chromosome 1 are together, both copies of the chromosome 2 are together, and so on. At the end of the prophase, the membrane around the nucleus in the cell dissolves away and releasing the chromosome. Mitotic spindle consisting of the microtubules and other proteins extend across the cells between the centrioles as they move to the opposite pole of the cells. As you can see in the figure that the spindle fibers are arising from the centrosome, and centrosomes are moving towards the opposite pole and extending the spindle fibers. Next is the metaphase. The chromosomes line up neatly end to end and along the center of the cell. As you can see in the figure that chromosomes are arranged at the center of the cell, and the chromosomes, every chromatids attach with the spindle fibers, and spindle fibers attaches to the chromosomes at the kinetochore region, and the kinetochore region. In the figure, you can see in the figure that the kinetochore region represents in the form of black dots, and the centrioles are now at opposite pole of the cell with mitotic spindle fiber extending from them. Mitotic spindle fiber attach to each of the sister chromatid. The next is anaphase. The sister chromatids are then pulled apart by the mitotic spindles, which pull one chromatid to one pole and other chromatid to other pole. Next is the telophase. During this phase, at each pole of the cell, a full set of chromosomes gather together. A membrane forms around each set of the chromosome to create the two new nuclei. The single cell then pinch in the middle to form the two separate daughter cells, each containing a full set of chromosomes within a nucleus. This process is called as cytokinesis. So, telophase is the process just reversal of the prophase. During the prophase, there is the disappearance of the nuclear membrane. But during this phase, the nuclear membrane start appearing. Next is cytokinesis. Cytokinesis is the division of the cytoplasm into two cells, typically begins during telophase. In the animal cells, cytokinesis occurs by a process known as cleavage. The first sign of cleavage is the appearance of the cleavage furrow, a shallow indentation in the cell surface. At the site of furrow, the cytoplasm has a ring of the microfilaments made of actin associated with the molecules of myosin. Actin and myosin are the same proteins responsible for the muscle contraction. The cleavage furrow deepens and eventually pinches the parent cells into two, producing two completely separate daughter cells, each with its own nucleus and share of cytoplasm. That's all. Please do subscribe my channel for more interactive and conceptual contents related to biology.